about, I guess, part of her PhD. The floor is yours.
Exactly. I'll have to find out how I share my screen though.
Well, you see, if you go under, you see a little green square and it says share.
Do
on
you
the lower
see?
panel.
I have shared it. Do you see the presentation?
No. No.
Oh.
I don't see.
Okay, let me go back to it. All right.
If you're done with your mouse, there should be a green box with an arrow pointing out. Did you see that? Yeah, it's coming.
Yeah. We see a June.
It's taking a while. There. Can you see now?
Yes. Great.
All right. Cool. So thank you very much for being online today. I would like to share with you my actual experiences from my PhD, during which I collaborated with about 200 people, I think, to carry through a European-wide multi-lake survey, and also the opinion we formed with several of you on how this actually type of surveys can be a good strategy for lake monitoring. I think this next slide is quite redundant for this audience because we're all lake experts and we work on understanding ecosystem degradation and loss of ecosystem strategies, ecosystem services. And we choose lakes because they respond quite sensitively to any environmental change. In order to understand lake, then if we manage to understand lake dynamics, then this could help us also define the scale and frequency at which environmental change occurs. But the problem is that monitoring all these 170 million lakes is not actually possible. So we need to make sure that we understand what kind of sampling frequency we need and what kind of scale we need to cover. There are several monitoring methods that we have available. At the temporal scale, we can either do routine sampling or automated high frequency, which are actually quite subjected also to the national funding, how many times we can visit our lakes, and also they're quite limited to study systems that are important for the regions. At the spatial scale, we have monitoring strategies such as remote sensing, and which is also, it can cover a large spatial scale and it does have the potential for temporal scale, but it can be subjected to weather conditions, and it's also not exactly open access, and it's quite dependent on algorithms and people that can understand how to extract the data from remote sensing. And the other method is disparate data, which we basically combine different data sets from different lakes to databases like Lagos. And the potential problem with that is data comparability, because most of the data sets that are collected are actually collected in a completely different way. The method that we are talking about here is the snapshot sampling. And what we define as snapshot sampling is the acquisition of biological, chemical, and physical parameters at intervals that violate the Nyquist-Shannon sampling theorem. So according to this theorem, to fully capture the phenomenon that we want, we need to sample at a rate that exceeds twice the maximum component frequency of the phenomenon that we want to study. So if, for example, we want to study diel reposition of algal communities in the water column, we need that they are subjected to short time scale changes. We need to actually sample at hourly intervals. Oh, I can't move mine. Okay, sorry. So we see that we realize that every method is every method is best for a reason. So what we need to start with is to identify what's our question, what do we want to get of this strategy. For example, what kind of scale do we want to cover? Do we want to cover a temporal scale or a spatial scale? And in this paper, we actually identify which strategy is the best for each scale and which ones have a potential to become good. Also, we need to know what we are able to invest in terms of time, money, personnel, and equipment. And if, for example, we want to go for a long-term effort, we need to make sure that we have enough money and enough personnel, and also instruments that they could be reliable for a long time. Then it's like to see what we can possibly get for the money, for the resources that we have invested. If it would be possible to integrate the data sets we get, what's the accuracy and efficiency potential, if we can actually use this strategy to have an international collaboration and share.
Yeah, I'm listening to the GLEON meeting.
Hello? Oh.
Yeah, I actually wanted to hear the one after this one, but it just started.
The thing has
Hello?
the microphone. And okay, and what would be the potential problems that we would face with this kind of strategies? In this paper, we do support that snapshot sampling is actually a great strategy because it needs a short-term effort and can cover a quite wide spatial, have a quite spatial coverage, and also provide good data integration and be efficient this way and remain relatively cheap. For the next slides, I will talk about advantages we get from multi-lake surveys. Multi-lake surveys usually follow standardized protocols that aim to minimize sampling effort per lake without necessarily sacrificing data quality. By this minimized sampling effort, numerous lakes can be sampled at large geographical areas, which is great if we want to assess the water status, the ecological status, and provide ecological understanding. There are several that they were led by PhD students. The one in South America by Costa et al., which aimed at understanding cyanobacteria occurrence at a latitudinal gradient that was defined by temperature. And then the European multi-lake survey, which was led by me in 2015, that aimed at understanding temperature and nutrient interaction in cyanobacteria, biomass, and toxins at a continental scale. Such type of surveys have also used at the national level, as we have on the two pictures at the bottom, which was the Nordic freshwater inventory of about 3,000 to 4,000, with about 3,000 and 4,000 lakes, and the National Lakes Assessment in USA in 2007 and 2012 that sampled about 1,000 lakes. These data sets have been used by several scientists to answer questions on food web issues, lake status, and lake status. Multi-lake surveys, as I said, they follow standardized procedures. And that's quite important to secure, because this way we can achieve a complete data integration. I'm bringing examples from the European multi-lake survey, where we organized a training school, where we brought the participants not only to teach them the protocols, but also to get the feedback on potential problems that they would face in the regions for sampling the lakes. And we try to integrate this feedback and get a protocol that would work for everyone. The second very important point was to centralize all key analyses, which means that they were done by one person on one machine to avoid having to do intercalibration between the different laboratories, and also minimize analytical error. This way, we see that multi-lakes can actually produce highly comparable data sets with uniform and synchronic data, and that actually makes the job of data curators easier, as they can manipulate the collected data better, understand where outliers can be found, and also perform better quality control, assurance and control. Multi-lake surveys are cost and time efficient. The lakes are visited only once during the sampling period, and that can allow for more lakes to be sampled. And also the cost is also minimized because the protocols are usually don't require expensive instrumentation. This way, researchers and institutes with different levels of funding and equipment can participate. And they are perfect for grassroots initiatives such as you usually do in GLEON, that they typically have limited financial means and rely on the motivation and dedication of scientists. In the European multi-lake survey, for example, to make sure that we all get an integrated water column sample the same way, we took the advice of Kale and Carey that suggested to create this, build this anaconda sampling device, which is basically made of a transparent hose pipe, a bit of cable, a weight to make sure that it stays vertical in the water, and a stopper. And this is actually a slide as a late answer to Mary Lofton's question during Oslo about how much did this survey actually cost us. And we see that for all the laboratory analyses that you see in the line from flow cytometry down to toxins, was about $15,000 for all the 350 samples we got, for triplicates, for replicates, for consumables, for technician time, and so on. And this cost was either covered from university partners that they had enough research money they were happy to pay the cost, or grants that we got from the European cost collaboration. So now down to what each data collector had to pay. I estimated that for sampling, the consumables for sampling one lake was about $20, while for building the anaconda device would be about $40 to $50. But then you have to consider that these are actually prices based on the Swiss market. So I would think that in other countries the cost might be less. And then finally, for express shipping between European countries, for a package of 2 to 10 kilos, the cost would be between 40 and 100 dollars. And that would also, I also included here the cost for styrofoam boxes and dry ice. Back to the advantages of multi-lake surveys. Multi-lake surveys actually follow a space for time substitution approach, where they actually study present day spatial phenomenon instead of long-term records that in many cases are unavailable. Here I give you an example, not a spatial to temporal direct comparison, but how a spatial dataset can actually show the same trends as you would get in laboratory experiments. On the right side, we have data that Pearl has collected on what is the optimal temperature for cyanobacteria growth, and it's between 25 and 30 degrees. And that was exactly what Costin found at a spatial lake response about cyanobacteria being in a higher biomass between 25 and 30 degrees. In order to achieve a good space for time substitution, we need numerous lakes, which is a drawback of this method, but it's possible if there are many collaborators. So numerous lakes, and the statistical power that we generate with these different lakes can overcome the risk that we usually get from long-term monitoring efforts in a few lakes. So we can aim for global lake response. And the last thing that we need to consider when we run a survey like this is to target environmental predictors that are very similarly at the spatial and temporal scale. Here I give you an example of what we did with a data set that we collected from the European multi-lake survey on understanding the continental scale distribution of cyanobacteria toxins in Europe. On the left, we see the toxin concentrations. The ratio gives the toxin concentration of MC for microcystine, SYN for cylindrospermopsin, and ATX for anatoxin, while on the right, we see the results of ordination analysis that show that mostly the direct effects of temperature as surface and epilimnetic, and the indirect effects of temperature on the water column stability as buoyancy frequency here, explain continental scale distribution of cyanobacteria toxins. We see that lakes with DMLR and DMRR would be mostly defined by buoyancy frequency. And indeed, if you look into other studies that were done, we see that deeper lakes usually with filamentous cyanobacteria would have DMLR and DMRR toxins, while lakes with shallower lakes where temperature and where epilimnetic temperature and surface temperature would be more directly affect the growth of cyanobacteria would be lakes with LR and RR, as we see. So we see that a data set like this can also inform trait-based approaches in ecology, where we aim at looking at not what the species are in the lakes, in our lakes, but actually what the species do, and how would basically, how by knowing our systems, we could understand what kind of reactions, what kind of like biological responses should we expect. There are several other multi-lake surveys that I'm aware of. Here I give you two examples. The one is FANACWA, that I believe several of you are part of. It's a global effort, and it's on fungal communities in lake sediments. I know that they have been using several, I helped them with several of the protocols, and at the moment I'm in collaboration with, in communication with Christel, because he's just drowning in samples. And the second one is in European and North America, that is about goniostemum. I'm not really involved with this one, but I have brought Raphael in communication with several people from the European Multi-Lake Survey Consortium to get some information what kind of, where he could find lakes that would possibly have goniostemum problems. Here is a paper that I've been talking about in case you want to get more information. And I wanted to say that EMLS was actually inspired by how things are done in GLEON. And it's another evidence that collaborative science has the power. So I really hope that you will continue this kind of collaboration. Thank you. Do you have any questions? I don't think I can hear anything.
Thanks for a great talk, Evi. Perhaps people could just speak up if they have questions.
Evi, this is David Hamilton. Mm-hmm. I just had a question on the use of both epilimnetic and surface water temperature. Mm-hmm. Aren't they just pretty much the same variables, except under times when you might get diurnal stratification? So doesn't that just reinforce the one variable?
Yeah, well, actually, this is a mistake that it actually should write metalimnetic temperature that goes all the way to the bottom of, the end of the thermocline region. So there was no correlation between the two variables. That's why we actually used it in the analysis. But you mean in a sense that would actually there is an overlap in the analysis, or?
Yeah, I'm just trying to reconcile surface temperature and epilimnetic temperature. But you indicate it's not epilimnetic, it's actually metalimnetic.
Yeah, it's metalimnetic, yeah.
Ah, okay.
Yeah.
Thank you.
Am I still sharing my screen? Because I don't know what's happening now.
You do actually, oh, sorry. You do actually the same thing as you did before, and then you say stop share.